Everyone, thanks so much for coming to the BLiSc Life Sciences Cluster Science Cafe. This is our 4th Science Cafe, and we're really encouraged by the kind of feedback that we're getting, and people seem to be enjoying this, so please keep coming, bring your friends, and so on. So today we have a really exciting speaker. His name is Mukund Thattai. He's the head of academics at the National Center for Biological Sciences. Earlier I said something about the BLiSc Life Sciences Cluster, so let me just explain a little bit about that. The cluster comprises three, I would not say institutions, 2 institutions and an incubating cell. So one is the National Center for Biological Sciences, and one is InSTEM, which is the Institute for Stem Cell Biology and Research, and we also have CCAMP, which is an incubating cell for the research that we do there. A little bit about Mukund. You'd be interested to know that Mukund actually is a physicist, but he studies biology, and his main area of interest is how cells are organized and how they function. I have to read from this because actually what he does is extremely complex and a little removed from what other scientists do. So he's an interesting anomaly in that way. Okay. So thank you all again for coming here. Please enjoy the session and welcome.
Thank you. So, since you did ask, it's too loud, let me see. Do I need to use the mic? If I shout, can you hear me? Which is better? Shout is okay? Yeah, it's better. Okay, shout is better. Perfect. Thank you. Okay, great. So we're very excited to have these talks in the city because we get to sort of meet people and understand why they value science. And it's absolutely our pleasure to explain what we do, but also I think it's quite important that people understand the science we do because we are a publicly supported enterprise. In some sense, what we do should, in the end, have some value or impact. And the impact may not always come from products or improvements in health. The impact can sometimes just come from a better knowledge of how the world works. So science is a sort of cultural activity. So what I'm going to do today is to convey to you some ideas about one of the central questions in biology, which is one of the defining features of living things, self-replication. Okay? Before we start, I'm going to play a game, and for this game, I need 2 volunteers. Raise your hands. All you have to do is to draw something. 1 volunteer, perfect. We need 1 more volunteer, very brave volunteer. 1 more, come on, we have 1. We have another, perfect. Okay, so what I'm going to do is to ask our first volunteer to stand back there, and you won't be able to see what's on this board. Okay? And our second volunteer will take one of these or many of these pens, and I want all of you guys to just name an everyday object. Anything you want, something that's pretty easy to draw. Could I have your name? Ira. Ira, okay, and your name?
That's a permanent marker.
So what Ira is going to do, use the A one.
That's a permanent marker.
Oh, that's permanent. Well, that's a good point. This is going to be a white pen. So what Ira is going to do is draw the object you guys suggest on this board. Okay, to start with. And then I'll tell you what the rest of the game is. So what kind of object shall we draw?
Glass.
Does it have to be animal?
No, it doesn't have to be animate. Any object.
A tree.
A tree. Okay.
Spoon. Notebooks. Cell phone.
Okay, so which one of these? Okay, so tree is pretty good.
Okay.
Yeah, tree is pretty good. Okay, so I'd like you to take up as much space as you want on the board and try and draw us a tree. So when you thought of these objects, what did you have in mind? You were just thinking about things that were in your everyday life or things that were fun to draw, sort of fairly obvious. Okay, let's see how this goes. You'll see. Okay, that's good. That's good. So thanks, Ira. Give me a hand. Now the game really starts. All Janvi knows, you can go back and have a seat. All Janvi knows is that somewhere over there somebody's drawn a tree. Okay, I want you, the audience, to help her draw the same tree on that screen, and she's not going to be able to, on that paper, and she's not going to be able to see this. Okay, so you need to make as close a copy as possible of what you guys see over there. I want you to instruct. You can use any verbal instructions. You can throw things at her, anything you want. But you can't see what's over. Okay. So what? Okay. So maybe I'll ask people. I'll draw a few. Okay, instruction.
Draw a trunk which are 2 parallel lines with 1 inch distance.
Okay, excellent. Okay. Very good. We have an engineer in the audience. Okay. Okay.
Good.
Very good. Very good. Anybody else? Who else wants to? Next instruction. Very good instruction. Next instruction. Yes. No, no, you can't use your hands. just verbal.
To show the rest of the tree, the foliage on the tree, draw something like a cloud.
Okay. So cloud. Okay. So we're getting the idea. Very good. Maybe this screen will be better. This is sort of a thicker marker so they can see it in the back. Yeah, that's fine. It'll work. Okay, good. So she's getting there. Remember, I want this to be as close a copy as possible to what's on that board. Okay, so we're getting there. But maybe already some stuff has gone wrong. I don't know.
Is there any detail on the trunk?
No. Okay, has she finished? Can she stop?
No.
No. Okay, what else is missing?
Branches.
V-shape. Oh, no. Okay. No hands. No hands. Just words.
Oh, the V-shaped thing.
No.
Okay. Okay, on the left of the cloud, just to the left of the trunk which coincides with the cloud, you need to draw branches.
No hands.
Which,
Okay, try and interpret that as best you can.
okay, the branch which looks in the cloud. Yes, the
Oh, good. You can also ask questions.
into the cloud.
You can also ask
Yes.
questions. Okay, excellent.
Okay, and then now you can draw another branch arising from the same branch that you drawn.
Okay,
Okay.
good. Somebody else?
Same
Anybody
thing
else?
on the right side.
Same thing on the right side. Okay, fine. Symmetric. That's
So
nice.
we have 1, 2, 3 branches. No, on the right of the. Oh,
We'll add to that.
Right. And finally, we'll do one last one. So I saw a hand right in the back. Mani.
I just said that.
Oh, you wanted to say same thing on the other side. So symmetry. That's very
Actually,
good.
I got something.
Yes.
Can you draw a Y-shaped branch?
Very good.
The
Where?
right,
Where?
a Y-shaped branch to the right.
Can I do a fairy?
No.
You don't know. You can't restart. No do-overs.
A Y-shaped branch on the right-hand side of the tree. Inside the tree, but on the right.
Inside the cloud.
Yeah.
Okay. So I think I'm going to call it maybe one or two last instructions. Yes.
Another thing, in the tree at the 11 o'clock position, draw.
Yes.
Yeah, in the
Very
tree.
good.
In the tree. Draw a reverse
An
tripod.
inverted tripod.
Tripod. Okay. Okay. Inverted tripod.
Yeah, right there.
Okay.
How?
Okay. Now we stop. Thank you. Thanks, Sam. So just so you can see what's going on here. Right?
Oh
No. I think you did very well. Okay, so this is actually remarkably close. Right? It's actually not bad at all. It's recognizably the same object. Recognizably an attempt to draw the same thing. Of course, some things it's not clear what the instructions were, but look, this is pretty good. Look at that. The Y shape was a very nice suggestion. This 1 inch also set the scale of the thing, so we didn't draw a tiny tree. So I think overall this is actually much better than I thought it was going to be. Of course, if somebody had wanted to draw a cell phone, it would have just been a rectangle and we would have been done with it. So I wasn't so happy with that suggestion. But some of the other suggestions also had enough intricacy that it required a little bit of transparent information. So that's, you can keep this. So, so now, let me get started with the talk. So that's the whole point. You see that it's not actually completely straightforward let's not even start with self-replication. It's about just replication is not a completely straightforward thing. And in order to think about replication, you have to actually try and do it yourself to see how hard it is, to see all the places where this kind of thing can go wrong. Okay. So now, apart from just drawing, I'm going to try and walk you through a collection of everyday objects where replication is sort of an appropriate way to think about this. And I want you guys to start to understand what all the different ways of replicating something are. And just so, just to get you guys thinking about it, here I have a couple of objects I just picked up from around the room. Here's a pair of things that look sort of similar. You can try and understand why they look similar. Was this built looking at that, and so on. Here's another pair of things that look similar. These are both, Chetan Bhagat books. Okay? They should be rather close to identical. You might want to think about this. Here's an object which I don't find another copy of in the room, but you might want to think about how to make a similar object. Okay? So, I'm going to walk you through, like I said, a series of examples, and you'll see that there's a certain pattern. Suppose I wanted to take a statue. Now, unfortunately, this is a small board, so I hope you can read it in the back. If you can't read it, that's okay. And I've apologize for my handwriting, but I have a statue and I want to make a copy of it. Let's say it's a big plaster statue, right? So we all know how to do that. We make a mold and then we make a copy. Yeah? And how do you make a mold of a statue? It's very simple. You take the statue, whatever it is, the mold is exactly the sort of negative space of the statue. And then you pour the clay or whatever it is into it and you get what it is. That's one way of doing it. So there are other objects for which you don't have a mold or a template. Now suppose I wanted to make a copy of the Mona Lisa. How would you do it? Using some sort of industrial process. So you have a painting. And these days you have photocopy machines, right? So you make a digital copy. And then you print it. Right? And this process these days, it used to be quite terrible, but these days you can actually take a painting, you can actually have an optical way to record all the information in the painting at fairly high resolution with all the colors, and then you can print out the painting on some medium at appropriately the right size and shape. Now what about something like a car? So if you go to a car factory, if you go to the Toyota plant which is in Bangalore, you'll see thousands of identical copies of the same car. And how do they all get identical? How do they get that?
Follow the parts are basically 3D set.
Perfect. There are parts. Yes. Somebody else said something?
Follow blueprint.
Blueprint. Excellent. Good word. Yeah. There are parts and blueprints. In fact, this is also how buildings are made, right? And buildings are made because you have a blueprint. You have sort of a small, scaled down version of what you actually want to make in the end. So in a sense, it's a bit like a mold, but it's not quite the same. Blueprint is on a different kind of medium, at a different scale, a bit smaller. But there are parts and blueprints, and together you make the copy. Okay, so then you build the copy. What about a cake? Right? Okay, recipe. Because ingredients, recipes. Right? And then you make. Now, are there any templates when you read these cookbooks? There's always that photo of the cake, and it's nothing like the cake that comes out of the cake. It's a bit like this pre-example, right? So somehow the recipe, which is what all you guys are trying to do now, the recipe way of making a copy of something is absolutely the most difficult way of making a copy, because you don't know if it's gone wrong. The cake itself can't tell you that something is going wrong. So it's a one-way process. So we have cakes and recipes. So I'm going to put, maybe I'll use this board to make a sort of spectrum of different ways of doing it. Right? So we have statues, molds, then we have paintings and photo. We have cars, buildings, we have blueprints. Then we have cake, we have recipes. And the spectrum I want to highlight is there's two different ways of making copies. One is based on templates, and the other is based on recipes. Okay? And any given thing will have a mixture of both these. But if you go out to the statue, it's mostly a template. You have a statue, you make a template, and the closer you are to the template side of things, the closer your whole copying process is to the object itself. Okay, so when you make a template of a statue, it's roughly the size and shape of the statue, it's made often of the same kind of substance. The photo of a painting, of course, is not quite the same size and scale, but in the old days of photocopying machines, you did have a film which was roughly the same size and shape as the object you wanted to record and print. Then you get to something like a car, which is some amount of template, because there are parts floating around, there is a door, it sort of looks like a door, and you want to fit them in a certain way, and some amount of blueprint. Okay, now IKEA has just opened up in India. I've never been to an IKEA store, but I'm told that you buy all these boxes and they're full of these bits of wood, and they come up with a blueprint. So you have some pieces and you have a blueprint. Again, like the cake, it never turns out the way the picture is on the box. And finally, you have the most difficult thing, which is these cakes and recipes. So in this spectrum of things, where does biology fall? So if a cell makes a copy of a cell, whenever you look around, you'll see identical copies of cells. Many of the cells in your body are the same as each other. Is that because they have a template? Is it because they're coming out of a mold? Is Is it because there's a blueprint? Is it because there's a recipe? What do people think? You raise your hand and count.
It's a mixture because
Mixture? Is it closer to the top or closer to the bottom? How many people think closer to the top? Okay, very good. How many people think closer to the bottom? Wow, okay, 50-50. So I'm going to try and convince you that the answer is actually not totally obvious. Okay, it's not obvious at all. And we're going to try and work through this whole question. Okay, everybody with me? Now there's a few other things which are cases which sort of don't fall in the spectrum, and I want you to keep these in mind. Okay. Again, I want you to think about if you go somewhere and suddenly see a large number of identical objects. Could they have arisen for reasons other than the ones I put down here? You go somewhere and you see a large collection of identical objects, as far as you can tell. Chemicals, excellent. So water molecules, okay? You can go look at a water molecule on Mars or on Earth, or in fact we can detect them on exoplanets optically, and we know that the water molecule on planets orbiting other stars is the same size and shape as the water molecule here. Is that the same thing? If so, why so? If not, why not? Large number of identical objects. So, were those water molecules templated? Were they made from a blueprint or a recipe? Why are they the same? The reason they're the same is there's no other way they could be. It's not a template. Okay, so the one last thing. It's a restriction of possibilities. It's restriction of possibilities. Right? The water molecule, despite the physics of the laws of physics, cannot fit together in any way other than this. Okay? So that's the difference between, say, a Lego box and some of these sort of battleship kits that we buy. The battleship kits, they go together in one way and sort of totally obvious. That is not self-replication by any means. Okay? It's not as if I were somehow able to change the size and shape of water on Earth, suddenly all the water in the universe would change its size and shape. Right? It's not looking to be copied, and it's not made from a plant. There's just one way it can happen. That's one piece. The second piece I want you to think about. So every day in the papers they publish these Sudoku puzzles. All the Sudoku puzzles at first look the same. It's a 9x. How many of you don't know about Sudoku? Everybody knows, right? It's this puzzle you play. It's a number puzzle. So there's a 9x9 grid and you have to fill the grid. All Sudoku puzzles start with the same blank 9x9 grid. But every Sunday, for example, some puzzle is published in the Hindu newspaper, and those people who can do Sudoku properly, by the end of it, all of them would have the same puzzle. They would have filled out exactly the same numbers, left to right, top to bottom. How come they all filled exactly the same puzzle? Because a few numbers were there to begin with. Okay? Now which one of those? So you've replicated in some sense the same Sudoku puzzle across thousands of homes across Bangalore. How was that replication happening? Was it based on a template or a recipe? Template?
Logic
Yes.
and elimination.
Restriction of possibilities, right? So it's not a template because you are not looking at a Sudoku puzzle and copying. Okay? It's not a recipe in the sense that you do this, then you do this, then you do this. What you do is a bit like what was happening with Janvi and Ira, where they were at least trying to get back some information. They were saying, you gave me this instruction, can I have some more information to do? So the way Sudoku works is because it's an implicit recipe. The recipe is not explicit, but comes with a whole bunch of constraints. No two numbers in the same row, same column, same diagonal, same 3x3 box. Those constraints are also part of the recipe. It's not a do this, do this, do this, do this recipe. Okay? In some sense, it's a restriction of possibilities. In some sense, it's a recipe. Again, biology uses all these possibilities. Okay? I just want you to think about all the ways this could have been done. Okay, so now let's stop with this. I'm going to leave this paper here so you guys can see it. Okay, it doesn't matter. Yeah, there we go. Hopefully that's cool. Okay. In all these cases, the thing in the middle is the important piece. Okay? Let that go, doesn't matter. You always start with something, go through a process, you come up with something at the end. The process requires machinery. It requires some human intervention. Right? It requires all kinds of gadgetry. But somewhere there's the formula, the recipe, the template, or whatever. So the question is how this template relates to the object itself, or this recipe relates to the object itself. I'm trying to convince you that templates are very obviously related to the object, whereas recipes are only abstractly related to the object. Okay? Now this is something we're all used to. Right? the idea of having information and that information being used to convey the object itself. It would have been very hard to explain this to somebody in, say, the early 20th century, right? Because it wasn't the way things worked. Things were based on blueprints or templates, but not on information. These days, it's very easy to talk to people about information because everybody has mobile phones, everybody has data plans, everybody measures everything in bits and bytes and so on. Okay, until Reliance Jio, but now, of course, we don't have to pay for data. But in the days when we had to pay for data, there was a certain unit of information. Right? So, anybody knows what a unit of information is called? What's the name of a unit of information? Byte, right? So typically it's slightly bigger than that, right? So you pay for gigabytes or megabytes, right? Or you buy hard drives that store information in terabytes. These are all units of storage of information. Okay? That is the unit which measures the size of this thing. Right? It measures the size of a recipe to make something. That's what those units are. Just like kgs is units of mass, right? Meters is unit of length. Bytes and kilobytes, megabytes and terabytes are units of information that you need to make something. Now, just as we played the game with the tree, we could have played the game where there was a human being over here and you were trying to convey to Janvi to make a copy of the human, right? But not using your hands because that's a bit like a template. You're trying to show how it is, but just using words. Okay? So, just using words, you want to convey how to make a copy of some human in this room. How much information would that take? If I could write down those words in a file and I could store that file on my laptop or on my thumb drive, right? How much space would I need to store that information? Okay, so I'm going to put this into context. Okay? So everybody clear? What we're going to do is to try and see, just like that tree building exercise, we're going to try and see how much information it would take to actually make a copy of a human. We're going to work this through. The answer will surprise you. Okay, so first a little bit of 20th century history. So we have figured out everything we need in order to answer this question. We haven't yet fully answered this question. That's where science is right now. So it might surprise you to know. So this is a very famous date in science, 1905. Anybody know why? Einstein. Yeah. Which of Einstein's theories is relevant to me today? So Einstein proved the existence of molecules. Okay? Einstein. Before Einstein, in the 1800s, molecules were an abstract idea. People thought that's how the world worked, but there was no proof that molecules existed. So many people don't actually know that the person who proved the existence of molecules was Einstein. In fact, he actually did experiments with sugar water. Right? So before this, there was a notion of matter could be a continuous object, but after Einstein, we knew matter was made in small chunks. This actually also connects to this whole idea of the quantum revolution and this whole aspect of physics, which has now become the basis of all of modern physics. In parallel, there's a very famous physicist, Schrödinger, okay, who, if you took a class in chemistry in school, and we all did it, we saw his famous equation. None of us actually understood what it was when we first saw it. And you heard about his cat, right? So he didn't actually have a cat. So Schrödinger, Einstein, Planck, Born, all these people developed a quantum theory. This was going on in Europe at a time when we went through 2 world wars, and due to the war, Schrödinger actually had to leave Europe and took refuge in Dublin. And almost exactly 75 years ago to the day, he gave a talk at Trinity College in Dublin called What is Life? Okay? Which is, in fact, the basis, the template for the kind of talk I'm giving you today. Schrödinger was a physicist, and he somehow got enamored with trying to understand how biology works. And exactly this idea, how do you make a copy of something? But what he came to the problem with was he knew that there was something puzzling about this. He knew that somehow all the information to make a plant or all the information to make a human can be compressed into a very small space. We know that, right? An entire tree, all that information is compressed into a single seed. And you know, if you toss a teak seed, you get a teak tree. If you toss an oak seed, you get an oak tree, right? So somehow all the information is in the seed. In the same way, all the information to make an entire adult human is in the fertilized egg. He knew the sizes of these objects. he knew how many molecules they contain. And so he knew the largest sort of file one could store in an object of that size. Right? You know, the bigger the hard drive, the more the information you can store. The bigger the storage medium, the more the information. So Schrodinger was very puzzled with the question of how you store so much information in such a small space. And he came up with some ideas. And his idea was that somewhere there's a separation between the object and the information storage itself. The information is somehow stored as a compressed recipe. Okay? And this he did just by looking at numbers and saying there's not enough information to store a little copy of a human in an egg, which then becomes bigger and bigger and therefore makes the big human. This is called a homunculus, for those of you who know. There was no space to make a homunculus, so it must be by recipe. Okay? Schrodinger's input actually was enormously influential to the 1953 discovery of DNA by Crick, Watson, Franklin. Okay, very confusing thing is DNA. You read about it in the papers, but very little about what you read actually conveys what it is. Let me tell you what it is now. So DNA is the little hard drive on which the information is stored. Okay, just like you have silicon bits on your hard drive, you have little molecular bits of DNA. Just like the bits 0 and 1 is how the files are stored in a computer, bits ATGC are how files are stored in DNA. So this, we went from the discovery or the proof of the existence of molecules to the discovery of the molecule that stores the information needed to make humans in this short span of time. Quite remarkable. In parallel, something else was happening in that part of the 20th century. Again, it's absolutely worth it. 1977, Alan Turing came up with the idea of something which eventually became this universal computer. Okay? Before Turing, you had different machines to do different computations. And Turing said, I could make a version of that machine. Any machine you want, I can make a version of it in my universal machine. I can sort of make a copy of it that will run just like a cash register, or just like a numerical calculator, or just like a log table. So that was Alan Turing. And he was everybody, I hope you know about Turing. Turing was the person behind the code-breaking efforts in World War II, which is the mechanism by which they cracked the Nazi codes, which was a big part of how the Allies won the war. Okay? So they came to this idea from code-breaking. Nothing to do with biology, nothing to do with physics. The other person who is relevant for this is 1948, a person named Shannon, who actually invented the idea of the bit. Okay? He worked at Bell Labs. He was talking about telephony, sending information over the airwaves. Again, part of the war effort is where this kind of information came out, code breaking, cracking codes. And finally, in the 1950s, there's a person very few people unfortunately have heard of outside of, a person named von Neumann, outside of physics. Von Neumann, he was a polymath, he was an enormously influential scientist. He participated in, among other things, the Manhattan Project. He participated in the creation of early computers. He also came up with this idea of a machine that makes a copy of other machines, but not abstractly in a computer, but in real life. Okay? We still don't have such a machine, but I want you to keep these things in mind. So, what I want to explain here is that science proceeds in sort of very unpredictable ways. People who were doing physics and biology came up with this version of doing things in a completely independent setting. People who are talking about coding, code breaking, and so on, computers, came up with a different way of doing things. And it turns out that both these are identical. Okay? So the whole formalism developed by Shannon, Turing, and von Neumann turns out to be exactly how biology works, we think. Okay? And this is extremely surprising. It's a huge coincidence. It's surprising in the sense that humans discovered both these things in the same century. We could have imagined another world where this kind of thing was known hundreds of years ago, and it took us much longer to make these discoveries. Or the other way around. By the way, if you have any questions, you can stop me at any time.
Yes, the whole structure of Science Cafe is supposed to be very informal and an exchange of information. So please, at any point in time, step in, ask questions, beat up the presenter,
Yeah.
throw banana peels or roses, but just jump in at any point. So just keep it fluid and just be very relaxed. And it's not a class; it's a cafe. So just.
And if you feel your voice not as powerful, we have a mic too. So
We have a mic as well. Yeah. Can people still hear me in the back? Is all good? Yeah. Question.
So now, after these many years, when looking back, we see
see the similarity of these?
It was evident actually right then. In the 1960s, when the genetic code was being broken by Hargobind Khorana and others, they really thought about coding, digital codes, and so on. Okay? Because these things did help each other out.
I see, okay.
Okay? So just 0 and 1 for computers, AT, GC for biology. Once we knew that, everything started to move.
Got it. So there's a difference here between the two situations. One is a discovery, the other is an invention.
Yeah, is it really an invention? That's what I'm trying to get at. Maybe there's no other way it could be. Okay. Okay, that's a very good point.
Yeah, because I was thinking, would there be a relation because we started discovering these in the natural life and then imbibing that into our things around us?
Historically, that's not how it happened, and my contention is any planet orbiting any other star where you find life will basically be based on these principles. If we're thinking about it, it's hard to imagine any other way. That was one Norman's contribution. But we'll get to that in the end. If life arises on another planet, what aspects of it will be similar to what we're doing? But now let's get back to the key question, which is self-replication. Before we get to self-replication, we're still doing replication. Okay? Now, like I said, to replicate now, you need a recipe or a template. Then you need a collection of machinery that will read the recipe and make the object. It's like the photocopier, you put the digital file, it will print out the object. Or the mold, where you give the mold and send it through the potter studio and you get the statue at the end. Or an entire car factory. And the important thing about all these cases is the same car factory from year to year can make different cars. You don't change the machinery in the car factory, more or less. You change the order in which you do things, you change the rough sizes, the timings. Like somebody said 1 inch for that tree. The next day you could make a tree that you said 2 inches. But you could still make a tree using the same paper and the same pen. Okay? So in all these cases, the machine that's making the final object is capable of taking in many different recipes and making many final objects. Okay? That's very important in terms of replication. If the only thing the machine could spit out was exactly one object, that would be sort of useless. Yeah? Okay. I don't know how many of you read these Kurt Wallander novels, but the protagonist's father, he paints this one painting his whole life. He keeps painting many copies of this painting. So that's not replication, right? That's just sort of one. Okay. So again, another metaphor. The thing that makes the object. You don't have to think of a Potter studio or whatever. Just think of, even now, already this is an old-fashioned metaphor, but think of a DVD player, which nobody has anymore. Okay? But whatever. So in the old days, you used to have a DVD player, right? And you get a DVD, and you get a DVD of some movie you like, put it in the player, and that's the movie you get out. The next day you can put a different movie in, you get a different DVD in, you get a different movie in. Okay? That's another metaphor for how the recipe for the movie is in the DVD. So I want you guys to guess how much information it takes, just like how much information it takes to encode a movie, how much information does it take to encode a human, and I want you to do this using the same units. Because as I said, DNA and computers basically use the same type of digital information. Okay? So for this, of course, I need to set the tone here a little bit. So a bit is actually a yes-no question. Right? It's either a 1 or a 0. That's what a bit is. It's a 1 or a 0 on a file. Usually the information that's recorded in computers is recorded in bytes, which are actually 8-bit bits. Right? Let's say approximately 10 bits. A byte is about 10 bits, or we can say a bit is about 0.1 bytes. Okay, that might be a better way to say it. So why did we have this very peculiar way of representing information? Because byte represents a piece of information that's quite natural to humans, which is what? It's a letter or a character. Okay. So we have 1 kilobyte, which is 1,000 letters, which is about a page, right? And then so on and so forth. You have megabytes and gigabytes and so on. 1 CD is actually, I think I have this right, 700 megabytes. That is, a megabyte is 1,000,000 bytes. 1 CD, okay? And 1 DVD is 4.5 gigabytes, which is 4.5 billion bytes. Okay? So how many songs can a CD contain? About 10, at most 20 songs, right? So you can work out that a song is about, say, 1/10 of this or 1/20 of this. Okay? How many movies can a DVD contain? About 1. Right? Okay, so a movie is about this big. How much does it take to record a photograph? A photograph or a song, you can say, is about 50 MB. A song or a photo. Okay. So in this spectrum, if I had a machine that could take the DVD that contains the recipe to make a human, we don't have such a machine. We do have such a machine, but I'll get to that in a second. Suppose we had such a machine, which is like your DVD player projects movies. You have a machine which you put in the correct information and it makes the correct human. You put your genome and you get this human, you put some other genome, you get that. How much information does it take to store that? Okay? So we have terabytes also, gigabytes, let's see. Megabytes is 10 to the 6 bytes, gigabyte is 10 to the 9 bytes, terabyte is 10 to the 12 bytes, and so on. So in this spectrum, how much information does it take to make a human? Is it as much as a song? How many people think you need just as much information as a song to make a human? No. How about just as much information as your favorite album? Okay. How about just as much information as, say, a Rajinikanth movie? Okay, 1 or 2. How about, 1,000 times that much information? How about 1,000,000 times that much information? Okay. So the answer is, it takes slightly more than a CD and far less than a DVD information to encode a human. Okay? So in other words, it takes less information to encode Rajinikanth than to encode a Rajinikanth movie. Okay? This is true. Right? This is the fact that should be astonishing. So the amount of space for a human genome is about 3 million bases. 3 million bases. A base is one of these letters, A, T, G, C. Since computer bytes are zeros and ones, it actually takes 600, so 6 million bits, or 6 million divided by 8 bytes. Okay, which you can see is far less than the amount of information we need to encode in a genome. In fact, how do you know I'm telling the truth? Is because these days you can actually pay companies about $1,000 to get your whole genome sequence, and they'll send back the information in a thumb drive. They're not going to ship this entire truck worth of hard drives and all that. But they can actually email you the information these days. So the information it takes to encode you is just on that little drive. Right? So this should be pretty shocking. It's shocking because even at the beginning of this, when we played that game, I would wager that we could have spent the whole day here trying to convey information about where to put that pen and draw the left branch or right branch and so on, and you would not have made the correct tree. Right? This whole process was sort of error prone. And yet, very reproducibly, your genome will make you, had you been able to run that experiment twice. The reason we know that is because identical twins have identical genomes and they make identical twins. To the extent that there's some variation, that's the amount of error there is. The amount of error between identical twins is far less than the amount of error between the template tree and the copy tree that we try to make today. Okay? So we have to think about how that's working. Are there any questions about this? So this is pretty astonishing. Yeah?
Very basic question. So when we speaking identical, are we talking about the physical attributes
Very
or personalities also?
No, so just the physical attributes, right? So once,
What was the question?
the question is when you say two people are identical and the identical information is encoded in the genome, is it just the physical attributes or is it anything else? So, so the fact is, of course, that your life experiences then govern everything else about it, right? And certain things will happen differently to different individuals, and that will govern what they become. In particular, there's not enough information in the genome to encode your brain, because that happens through an engagement with the environment. Okay? But you know this because, again, identical twins don't have identical personalities. So look at twins, that will give you a very good idea of how much sloppiness there is in this whole process. So we've got now the idea of how much information it takes to encode a human, which is much less than a DVD, just a few CDs. Okay. So,
Can you please be a little louder? You're not very audible at the back.
oh, sure. I'll try and project. Okay. The mic I find to be a little. I'm going to trip all over it. So, now I said we have a machine that can take genomes and make living things. Okay. Yes.
I'm sorry. When we say that all we need to know is the basis to encode a human being, what about all the knowledge that is required to replicate this basis?
Very good. That's exactly what I got.
Out of it.
Thank you for that question. Right. So this is roughly the same as me going back to the Stone Age and handing somebody a DVD and saying, here, go watch a movie.
Yeah, that would work, right?
Yeah, it won't work. Because what don't we have? We don't have the DVD player because we don't have the electricity and so on. But the main thing is we don't have the DVD player. Okay? So remember what I said. For the moment, I'm only talking about replication. I have not yet talked about self-replication, which is what your question is sort of getting at. And I will answer that question in just a little bit. So what I want to do is to have a machine where I can put in a genome and I can get out one type of organism. I can put in a different genome, I can put out a different type of organism. Okay? I have such a machine in the lab. Okay? It looks like this. So there's a beaker, and in this beaker there's sugar, salts, many types of salts, and water. Right? And it's all kept at some room temperature or maybe body temperature. Then I have a pump, and I pump it into a black box. Right? So the liquid is going into this black box. Then I have another pump. I'm not telling you what's in here, but that's my magic machine. I have another pump, and that gives you this output flask into which this stuff is dripping down. Okay? So we have this in the lab. This is exactly how it looks. There are two pumps, sugar, salt, and water. You have to keep replenishing the sugar, salt, and water, but you can just do this very easily because you can go dig this out of the ground. You can in fact get these ingredients from asteroids if you wanted to. Right? So basically, as far away from life as possible, you can go collect all this stuff. You can make this thing. Yeah.
Where do you put the
genome? I'll tell you. The genome goes in here somewhere. I have a little computer here. Somehow I type in the genome, okay, and I have a process of doing something to that black box.
So what is the output there?
Good. So initially when I run the machine, what do you think is going to happen? Sugar, salt, and water goes in. So sugar, salt, and water comes out, right? Yeah. It's just a way of transporting material from the left to the right of the page. Then I do something. And sugar, salt, water go in. Living cells come out. Okay? I have such a machine in the lab. Sugar, salt, and water goes in. Living cells come out. And let's say, I don't have humans coming out of this tube.
When you're creating life,
I have living cells coming out of the right side of the
you're
tube.
creating life.
Let's talk about it. What have I done here? Okay? And what cells? Bacterial cells. As it happens, the one that's in the lab is bacterial cells. Can anybody name a type of bacteria? E. coli. Anything else? Salmonella. So, for example, I can have E. coli coming out, and then I can do something on this computer, and then I can have salmonella coming out.
But you have some people like somewhere.
I have this computer.
You have only.
I have this computer.
You only a thumb drive.
So I have this computer, I have some, and then I have cells coming out. Good. So this should astonish you. You should maybe accuse me of lying or something, or at least yeah. Okay.
I agree.
Okay. So it should astonish you. And the fact is that this astonishes me every day. But let me then tell you the answer, and then the answer you will say, how so obvious? But it shouldn't have been so obvious. But remember your astonishment now, and try and preserve that astonishment after I tell you the answer. So obviously, initially, sugar, salt, water moves from left to right. After I do something, sugar, salt, water goes in, living cells come out. What did I do to the box? What did I add in there?
Give instructions.
Some bacteria.
I added one living cell. Somewhere in here, I put a single living cell. Right? I took a single living cell. That single living cell is what? That's my DVD player.
It didn't come from a computer.
Not yet. My full setup is very interesting, right? I can change the species.
Are you changing the genome of the species?
I'll tell you how I do that. But let's do the first bit first. The way I made the sugar, salt, water transportation device into a life creation device is with the addition of a single 1 micron. The wavelength of light is about that much. The wavelength of visible light is about that much. So something just slightly bigger than the wavelength of light. I took a single living cell. This experiment has been done. Put it into this black box. I didn't tell you. Then what happens? It takes sugar, salt, and water. And what comes out of it? Living cells of the same
type.
species. Okay? So here, the information, the DVD and the DVD player are the same. It's like a DVD player with the DVD is stuck. You can't change the movie. It's a whole DVD player, but it's only playing one movie. Okay? Got it? Now, I can type a new bacterial genome onto a computer. I can go to a company that's in somewhere in Bangalore, they can synthesize, well not in Bangalore, but currently, let's say in San Francisco, and they can synthesize the whole genome. This is true. Chemically, using only non-living processes. Okay, there's nothing in principle that prevents this from happening. That genome is shipped to me in a little tube, looks like transparent material. I can inject that into this DVD player, right? And the E. coli DVD player will start to play the Salmonella movie. So the bacterium that comes out of this will be Salmonella, not E. coli. It's been done. Craig Venter did this exactly. Okay?
You're getting from
Not with these species, but yeah.
you're getting from your genome sequencing not
They give me the DNA. At some point, I have to interface with the right materials.
the create DNA,
They create well. DNA is just chemicals.
but it's a very long chemical.
No, each piece is very simple. I just have to string them together in the right order. Right? ATGC, very simple. You know, you've heard of ATP. The ATP is the A in the ATGC, which is something I didn't know until quite recently. But the adenosine in the DNA is the same as the stuff that gives you energy. Right? So these small molecules are everywhere. They're even in space. Okay? You just have to. The company just has a machine that puts them together in the right order. So I order that. I put it in. So now, instead of the DVD, I get a genome. Instead of the DVD reader, I have a bacterium. Now, so far, the experiment has not been done where E. coli has been converted to Salmonella. It's been done with a different type of bacterium, but has been done where a bacterium of type A has been converted to a bacterium of type B, using type A as the DVD reader and type B genome as the DVD. This has been done by them. Question? Excellent. So it's actually quite difficult to stuck out the genome, but if you put in two genomes, once the cell divides, one of them accidentally gets one or the other, and then you select for the right one. Yeah.
This is a type of life creation, right? Because you're taking a non-animate DNA
I can't do it without a living thing in there. I can't do it without a living thing in there. Yeah.
You said type A and type B, we didn't type B. Can't it be type C?
It'll be whatever I input. Whatever I input. It'll be whatever I input. And I can also modify its properties. I can make it orange, green, I can make it big, small. I can do all that on the computer, and I can get those kinds of things. Okay. So this is now we're coming very close to self-replication. We're still not at self-replication. Where we are now is, I have a living object, just one. Okay? Then I can make any number of genomes. Then I can put it into that object. And that object doesn't project a movie. It actually does something much more subtle. It makes another copy of an almost identical object out of itself. Now in the engineering world, what would be a metaphor for this kind of thing? Okay? So remember I put up that thing about templates and recipes, right? So one of the things I said was, if you have templates tend to be roughly the same size and shape as the object, whereas recipes tend to be compressed. Right? There's another thing which is slightly different. Right? So if I wanted you to make, that's why I pulled this thing up. If I wanted you to make a template for this, you could probably do it as a mold, but it would not produce a musical instrument. Okay? You could take a photograph of it and it would look exactly like this, but it would not produce the right sound. How would you actually make this? Right? The materials need to be correct. In other words, to make this, it's not sufficient for it to look correct. It has to be correct right down to the molecular level. The wood, the string, the metal, everything. Okay, at the atomic level. So if I gave you this and said make a copy of it today, there's only one way to do it. You have to take it to a craftsman, the same person who made this, and he can replicate it in a manner which is very hard to write down on paper. Can a 3D printer make a musical instrument without significant additional input? That's the question. Okay? But I'm glad you brought that up. So that's precisely the point. Today we have things called 3D printers. 3D printers in my mind are just one dimension up from a photocopying machine. What do? They take a digital file and instead of printing it on paper, they use various types of polymers and they use all kinds of jets that move around and they make an object. Right? But I put it to you that right now you can't have a recipe where a 3D printer spits out a guitar without significant intervention from a craftsman in the middle. Okay? So the difference between replication and self-replication is, among other things, the materials themselves have to be the same all the way down to the atomic level. Right? So instead of the DVD player playing a movie, it will be something ridiculous like a DVD player somehow spitting out another DVD player. Current human technological capabilities have not reached that stage, but we're very close. The day somebody makes a 3D printer which can use generic stuff that's floating around in its input tank and somehow make a copy of itself, then we have something that very much approaches von Neumann's version of a self-replicating machine, which is what living self-replication actually is.
There's been a 3D printer, I just read recently,
Which can
simply, it's made components for itself, and which later you assemble.
which you assemble in somewhere.
But it, can't manufacture assembled 3D printer.
Right. So now we start to look at the fine distinctions between, suppose tomorrow somebody comes up with a 3D printer which you pour plastic on one side and a few bits of metal and it makes a full 3D printer on the other side. At what point you say now it's self-replicating, right? So this is a very good question, and this is the kind of question people are wrestling with now. Just like in the artificial intelligence world, they're saying now that machines have this amazing ability to read and recognize image, at what point do you call it the same as human intelligence? The same way for self-replication, once we build an object, at what point are you going to call it a self-replicating apparatus? Okay. Now of course, and what's going to happen eventually is they'll say, yeah, of course 3D printer can replicate, but life does this other fancy thing. Right? So now let's discuss what those other fancy things are that life does, which is not the same as what a 3D printer replicating itself does. One thing is, living things don't just wait for the materials to come to them. That's, they actively go out and find the right set of stuff. So if you think, sort of eating food and making more of yourself is sort of an obvious thing. You can't just take a bunch of garbage, put it in your car's petrol tank and expect the thing to sort of move. It doesn't work that way. But living things have the ability to look in the environment, find the right stuff. If the right stuff isn't available, to modify the stuff until it comes in. Again, I want you to think about how many of you, so these days everybody in Bangalore lives in apartments, right? How many of you have like a balcony garden? We have a balcony garden, right? So when we first started that, we had a bunch of pots and we had a few seedlings, right? Of course, if you've been doing that long enough, eventually you have this verdant garden on your balcony. At the end of that process, after several years, that balcony is actually much, much heavier than it used to be. Where does that extra weight come from?
The air.
From the air. Right? Your balcony actually became heavier because the little seedling, in fact the seed, started to slowly accumulate carbon dioxide from the air and incorporate it into every molecule of itself. And atom by atom of carbon, your balcony became heavier. Okay? That's the other thing life does. Right? And this is pretty astonishing again. Okay? So, living things do that. And finally, living things do one other thing for which I don't have time to talk about today. It's beyond the subject of the talk, and I'm about reaching the end. Living things make imperfect copies. You don't make perfect copies. And this is crucial. Why? Variety, diversity, evolution, natural selection. If there was no variation, there are no better solutions to problems. Okay? So it turns out that self-replication is astonishingly good, but not quite there. Right? So even bacterial cells, every bacterial cell in its genome, in the tube that I have in my lab, will have at least one mutation. The genome is a billion bases, and there'll be sort of 1 per million, or maybe 1 in a billion. But 1 in 10 bacterial cells will have some mutation in its genome. These mutations are just changes in the file, the computer file, to make the bacteria. To make imperfect copies is actually a really, really clever trick. Think about it. If I go to a car factory and I mess up one page of some blueprint, you're not going to get a sort of different car which is still a car. You're going to get a pile of junk that doesn't work. Okay? But computer scientists have thought of ways to do this. So it turns out that when you take photos on your phones, every time you take a photo, there'll be some error in the way in which it's stored. If it's communicating with the cell phone tower, there'll be some error in which it's transmitted. Always there are errors. And computer scientists have figured out ways for the files to detect the errors and self-correct. So you can correct most of the errors that occur, but not all of them. And eventually you make an imperfect copy. That imperfect copy is a substrate for evolution. Okay? So now I'll finish with the final metaphor.
Question.
Yeah.
That topic. So that when you put the analogy between computer scientists and physics biologists, computer scientists you mentioned have error correcting codes. Is there a similar mechanism
In biology,
Yeah, like if you take
this is what we study. This is what we study. So there are error correcting codes up and down. What Schrödinger actually pointed out in his lecture 75 years ago is he knew how much noise there is from just temperature, thermal fluctuations, and he knew how much noise there would be from quantum mechanics. So he knew that anything below a certain size will constantly be buffered and changed. Right? Even on your hard drives, once you make the bits too small, in fact, we've hit the end of Moore's law. Now, the bits are too small, there's too much temperature, things are constantly going. Once things are too small, they just flip all the time. Okay? So somehow DNA, ATGC basis, they constantly, A changes to T, to G, all these things keep on happening. And there are layers upon layers of error correcting machinery where you don't see it on the surface. It's correct, not just at the level of what's recorded. In fact, I didn't finish that whole piece, right? But let me say, right? The DNA is just the substrate. Right? The DNA is like a page and some ink. Right? The actual sequence, which is A, T, T, G, C, whatever it is the information. The magic of modern information theory says it doesn't matter where the information is stored. It can be stored on DNA substrate, silicon substrate, whatever it is. So what biology has done is it separated the actual information, which is the sequence, from the substrate. This is a manner of saying it's a recipe. There are error correcting mechanisms also at the level of implementing the recipe. Right? So you could mess up the blueprint and you could say, oh, there's an error in the blueprint, I fix the error and then I make it. You could also have a factory where as the production line goes, you implement corrections. Now, typically that doesn't happen in an industrial setting too much, but it happens in the context of crafts all the time. I want to make a guitar, I get some wood, I start working. Right? And you don't just make it, you check, you do this, you do that, there's error correction happening all the way through. So biology works that.
These error correcting mechanisms in genetics have been discovered?
Some of them have. Some of them, like Hopfield discovered one called kinetic proofreading in the 70s. And now we've discovered many more. Okay? Fine. So the last thing. So I'll close with this and open up for questions. Whenever I give these talks, so unfortunately the way biology is taught in schools, none of this actually interesting stuff comes out. And you don't have to reach very, very complicated parts of biology to reach questions to which we don't know the answer. It's part of what I've been trying to convey. And the problem is sort of a confusion between the details and the big concepts. So you don't need to be a detailed person to do biology. We need details to understand lots of examples of how things work. And then we need to pool all those examples to come up with some big idea. So I've often found that this idea of somebody adding a cell to the black box, when I tell people that, they say, oh, you should have said that, then it's obvious. Then one cell makes 2 cells, make 4 cells. Right? So the idea that something initially was astonishing, and suddenly I tell you one little thing and suddenly it becomes obvious. That can't be, right? If it was astonishing, it is always astonishing. Even though in retrospect, it looks obvious. But I found a much more sort of, the metaphor sort of is much more disconcerting to people. Right? Which is my bag of atta. Which is what I brought up. So of course I brought up atta because, AT, GC. But actually, there is something animal. So you go to Food Wars and you buy a packet of atta. Right? We've all done this, right? If you open it that day, you can make your rotis and everything is fine. But if you forget about it in the back of your pantry, you come back 6 months later, then this is what? Is it a bag of flour or a bag of flour beetles? Or bouillon. Right? There'll be hundreds and hundreds. If you're lucky, maybe thousands, right? Okay. So just think about this. My question is, I'm not going to ask this deep question of when did something go from being non-living to being alive. The question is, when did this thing go from being vegetarian to non-vegetarian? Okay. And the reason this is an interesting question is this bag of atta is sealed at the factory. No air or water gets in or out. No material gets in or out. But all there is at the beginning, as far as you can tell, microscopic detail, is flour, which came from a living source, but as it is, it's dead. It's a non-living substance. A lot of carbohydrates and so on. And it's certainly vegetarian. At some point it becomes flour beetles, which is certainly not vegetarian, although it's quite healthy. In fact, most vegetarians in India get their trace vitamins and so on from insect residue in flower and fruit. Okay? This is true. So, look at what happened. A flower beetle. So, this is not like a bunch of random mutant flower beetles that all look different from each other. These will all be flower beetles of the same species. And to a non-expert, they look identical. But you look at a go down in Bangalore and then you go look at a go down in Mysore, you might have a different species of flower beetle. Right? So, this is like my black box experiment happening in your kitchens every day. So, why do I get a particular species of flower beetle in Bangalore and there's a different species in Mysore? Must have been a difference. What was the difference? Conditions were in the warehouse before the packaging. So, if you go to these warehouses, I'm told, just piled up grain. And what's on the grain? Insects. Right? But what was in this packet? It wasn't just brown flour. Must have been something else in there, right?
An egg or
An egg? At least two, because these are sexually reproducing creatures. There must have been at least one male and one female. Okay? This is like your Garden of Eden for these. Okay? But anyway.
Or the bag itself.
The bag itself. Yeah. Well, sure, but somewhere in there was a single or a few handful of copies of the genome of this beetle. Okay? And it's sitting in your pantry, in the back of the cupboard. Now imagine what is happening over a 6-month period. That single genome, the microscopic object, has initiated an atomic level rearrangement. Every atom in this bag, okay, it's broken this bond, put it over there, broken that bond, put it, and slowly, piece by piece, it's built up one flower beetle, then another, then another, then another. That's what it's done. Right? The astonishment should come by just looking at the initial and final state. Don't get caught up inside here. Of course, you had the flower beetle, it ate some food, it made new flower beetles. Because at that level, biology becomes very everyday. You lose what makes it special. What made it special is a single microscopic copy of the genome. You can't even see it. Somehow influenced every atom in the packet. At some point, it became from a vegetarian packet to a non-vegetarian packet. At the atomic level. That's the astonishing thing. That's what life is. Now, the biggest packet of atta we know, of course, is the entire planet. Okay. We started non-living stuff. Some point, somewhere, we don't understand fully. A self-replicating system arose in one little pond somewhere. And it has totally transformed at the atomic level, at least on the surface, the entire planet. Right? That's the infinite power of self-replication. Thank you. So, as Mine said, not meant to be a lecture, although it went into lecture mode a little bit, but questions, be happy to take questions. If some people have to leave, I won't be insulted. It's not a synthesis of life.
You'll have to repeat the question.
The question is, I'll repeat the question. The question is, in that black box in my lab, I still needed one living organism. So I haven't synthesized life. I never claimed to synthesize life. So far, we haven't been able to do it. But let me say one thing. It's possible in the next 50 years that in some laboratory, in a painstaking way, somebody might actually create a cell. Okay? It's possible. A cell which can self-replicate and therefore has all the attributes of life, right? But which did not come through a living process. It's in principle, because after all, it happened once. It happened in the past, right? So it's possible. My response to that will be, it's a bit like, making a copy of the Mona Lisa and then claiming that you invented it. Once you already have a living process, then by just copying it's sort of a lesser achievement than coming up with a totally independent mechanism that also achieves self-replication, imperfect replication, evolution. Okay. But it turns out we've done that without knowing. So, computer viruses and these kinds of things arguably are a sort of form of digital life, and we can discuss that later. But in the physical sense, even if someone were to invent a cell in a purely chemical way, I would not count that as a big achievement because all you're doing is copying the existing cell. Okay, Tom.
Yes.
So, technically, the bag of atta
is not a vegetarian thing. Just because we don't see the genome, it is. It is. Don't worry, it is vegetarian. Mostly it's vegetarian. It is vegetarian. Yeah.
Could you brief more about the black box? How exactly the process is happening?
So actually, the black box is back here. Yeah. So the black box. There's nothing in here. It's just another flask. It's just another flask which has liquid.
But how are you inserting the?
I think that's the question. How are you inserting
the genome?
Chemical
Yeah, okay.
DNA?
So you can actually get a chemical genome in a little Eppendorf tube, like so, about that size. You can actually take it. You can electroporate the cell, so it has sort of holes in its membrane. Cells naturally take up DNA, and then it will fix itself, and then you have a cell with that genome.
So,
Totally routine.
the membranes have their holes.
Yeah. So it turns out bacterial cells are constantly looking for other DNA to take up, right? Because that's how they. That's one of the modes by which they evolve.
So will there be, let's say, because the bacteria now has two different genomes, its own original and a new inserted one, will there be conflicts
Yeah.
or maybe even mutations among them?
Yeah. So this is a very good question. So if I could do this, if I could take E. coli and make it to Salmonella and then make it into something else, eventually could I get a woolly mammoth on the right side? In principle, I can, right? Because it did happen in real.
But what is the biggest? Is the human
Bacteria. But the point is very important. So these days people are talking about something called de-extinction. Okay, where they're actually talking about bringing back the woolly mammoth, by finding its genome, putting it in an elephant egg, and then implanting it in an elephant uterus. Okay? So the fact is, it's just like these sort of North American DVD players and Japanese DVD players use different formats. Not all DVD players are built equally. Just taking a genome, putting it in a random cell doesn't mean it will be able to read it. Okay? So you have to take it to something close, and then close again, and then close again, and so on. And so there are problems. But there is something quite astonishing. You can take a gene from a deep sea jellyfish, it's called GFP, it's a very famous protein, called the green fluorescent protein, it comes from jellyfish. And you can actually insert that gene into an E. coli cell, and the E. coli cell, the reading mechanism will happily read that gene and carry out those instructions. And it will make a green fluorescent protein. Right? And the jellyfish and the E. coli haven't had a common ancestor in about 2.5 or 3 billion years. So the level of interoperability of biological digital information is much higher than for human digital information. And you might want to think about why all cells somehow retain the ability to read at least parts of the genome of other cells. It's mysterious.
Would it then be a new species where you actually...
This is just semantics. It's not an interesting question.
Is there any possibility, like if we turn E. coli to Salmonella, at some particular point of replication at a particular situation, can it turn back into the E. coli?
So, once you've lost the original information, once that genome is totally gone, it would be chances 0 of reverting back. However, if you just change one base from A to T, then it might change back from T to A in the next cell.
Like cell repair process is there, DNA
Where, good question. So I took my E. coli, I put the Salmonella in the genome. Now it's been Salmonella for 100 generations. Right? Now you're saying somewhere the repair mechanism. How will the repair mechanism know it is meant to be E. coli? Where does it look up the recipe for E. coli? And then fix it. It doesn't have all that. It's not another layer of stuff, right? It is what it is. So it can't repair itself because by now it thinks it's Salmonella.
In a way, we can still call the black box a machine with the cell because the cell is a machine, which is replicating.
Black.
Yeah, he said the living things make imperfect copies of themselves, and it's actually a clever thing to do. Is there a little more elaboration on that?
So natural selection, Darwin's. So here's a quiz question. So the unit in which you measure imperfection is called the rate of error per letter per generation in DNA. Right? So our DNA has 3 billion bases. E. coli DNA has 4 million bases. E. coli replicates every 20 minutes, and we replicate every 20 years. Okay. Now you can ask, what is the per base per generation error rate? Now, how many of you think the error rate is much lower for humans than for bacteria? Per base per generation. Okay, 1. How many of you think it's much lower for bacteria than for humans? Per base per generation. Even though bacteria, of course, reproducing much faster, but per event. So it turns out that bacteria have much better error correction than humans. Okay? But not for any reason. It's just that we can get by with this amount of error. That's all. Okay? There's nothing in principle that prevents us from having, these days by the way, these days you can actually interfere with the DNA replication process in human cells. You can actually correct errors because as you were saying, I don't know if I were an E. coli converted to a salmonella how to fix the error. But I know what my genome is supposed to be. And if some cell of mine has a mutation, today in principle it's possible to fix that mutation. Because I have a copy of my genome on a USB drive. Right? So these things are possible. But we do continue to evolve, yeah.
Can we have that same process done using stem cells other than the bacterial cells?
So stem cells is just one type of cell and not particularly different or interesting.
So because they can replicate into any other cell.
Yeah, so this is actually not true. So because stem cells, you hear a lot about stem cells in the news, and a lot of that reporting is a bit overblown. Stem cells are just one of many types of cells in the body. It turns out that every cell in the body came from an egg, right? So we know that one type of cell can become another, become another, become another. Some types of cells stop growing. Right? In a multicellular organism, there are two types. So stem cells are not a magic bullet for anything. They're just a type of cell that can continue to grow and develop. There's nothing mysterious about them. Right? In principle, you can certainly use with stem cells, but what you would do with that capability is a different question. Whether you want to use it for therapy or whatever. But remember, as I said right at the beginning, don't think of biology just as a way to fix health problems. Okay? That's not what biology is. Biology exists to transform planets. That's what biology is. Yeah.
As science managed to figure, how would you compress that much information in say an egg or a seed?
Yeah.
Because
Good. So I didn't bring back this thought. I was hoping somebody would ask. It is astonishing that we need just a few CDs to encode the human genome. So biology has learned to compress the information very well over a long process of evolutionary time. Part of the way that works is restriction of possibilities. So it turns out with just a few of these pieces. So remember the Sudoku puzzle, I said I put a few numbers and the rest sort of falls into place. In the same way, in biology, I set up the initial condition and then the laws of physics take over. And that restriction of possibilities is what leads to a human. Okay? In a very computer science way, let's say this book. Any other book of the same size, I know exactly how much information I need because each letter is a byte. I can count how many letters. If you count how many bytes, there will be some large number of bytes. Each byte could be, or each bit, let's say, could be 0 or 1. The total number of books I can encode is the first bit could be 0 or 1, second could be 0 or 1, third could be 0 or 1. Okay? And the total number of possible books is therefore 2 times 2 times 2 times 2 times 2 to the power of the number of bits I use, which is pretty large. To compress information, I don't have to encode everything about the object. I just have to say how many possible humans are there, ever in principle, which one of those is you. Right? So when I store a picture, the way a computer stores a picture is not by showing the screen how to draw the line and so on. It's just there's a space of possible pictures which is restricted. And this particular picture is picture number 6,571 of that. So we lie somewhere between the amount of information you need to make something from scratch and the amount of information you need to make the collection of possible humans. I know it's not a very full answer to the question, but it's because of restriction of possibilities. I can discuss this later.
I read about 15 years ago that smallpox virus was created in the lab.
Sure.
So was that done with completely non-living?
Sure. A virus is just a piece of DNA or RNA.
It has a protein.
So that's okay. You can actually make proteins in the lab.
When they said that they kind of did it in a factory, not using a...
Today it's possible to do it in a factory without any living. And that's fine. It's not very, it's technically challenging, but it's not conceptually very complicated. The virus is not a living thing.
Virus is not, it does replicate. It does.
No, not on its own. The virus is the DVD, and it needs a DVD player in order to replicate. Okay.
You said it is some other
Planet.
planet.
Yeah. What would life look like on other planets? So for
You
me,
said
life.
it would be similar.
So for me, life on other planets would have three features. It have three features. So life on other planets. You would have information. So I think it's sort of reasonably clear. This is what von Neumann said, that you have to somehow separate the recipe from the object in order to make reasonably complicated things. Okay? So that means you have to use information to compress it. Secondly, there must be evolution. Right? You might invent one type of thing, but if it doesn't keep on changing, it's not going to become more complicated or invade new niches. And there's a third piece which I have no evidence. Obviously, none of us have evidence for any of this, but there's a third piece which I think will be common to life on any other planet, which is something like a cell. All life on Earth is made of cells. A cell is a sort of legal contract that allows you to capture your own innovations and prevent others from using it. Right? And so evolution, in the absence of this kind of legal setup, eventually ends up people keep stealing each other's ideas and things reach sort of minimal level. But what cells manage to do is they manage to privatize information. And so their own innovations are a benefit only to them. And it's an amplifier of evolution. So cells, information, evolution is what I think any planet will have life that has features that are not identical to this, but have sort of aspects of all these. Okay. Great. So thank you for your attention.